Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we'll be talking about the Qiime analysis pipeline. Qiime is one approach to dealing with amplicon sequencing data, and you can also use it for metagenomic sequencing data. And it was initially developed to help analyze multiplexed microbiome data, where you have a whole bunch of samples that are mixed together with different barcodes attached to the beginning of the reads on the different sequences from those samples. So you do that, mix them together, run them on a sequencing run, and then you have to, it's called demultiplexing, or separate them out afterward based on their barcodes. So Qiime will do that, and then it will process them, do quality control, and do many of the common types of analysis. So it will basically get you to the point where you have sequences assigned back to their original samples. It'll build phylogenies, assign taxonomy, do all the diversity analysis, and then it'll generate figures and help you do statistical tests. There's predictive modeling, lots of other features. So the general approach for Qiime, the kind of core analysis, is that you start with these tagged sequences, sequence them, run a particular script from Qiime to demultiplex. You trim off the barcodes, then you use those sequences to build a phylogeny telling you how they're related to each other, or you can just compare them to reference sequences and then you put them in the reference phylogeny. You assign taxonomy to them, and then you do all of the diversity analysis. Here's the Qiime workflow in a flowchart, and we'll be referencing this throughout the semester. I should mention there are plenty of other ways to analyze microbiome data. The mother pipeline is another very popular approach. They both have pros and cons. We'll be using Qiime for most of this. One nice thing about Qiime is that it lets you try a bunch of different methods at each of the points of the analysis. So much of the functionality in Qiime is actually written into Qiime, but it also serves as a wrapper for many other tools. So if you want to do OTU picking, for example, in Qiime, you can use uclust, summaclust, swarm, cd-hit. You can actually use mother and several other different approaches. And Qiime knows how to talk to all of those different programs, so it makes it very easy to swap in different choices during your analysis. Key files that you need if you're going to be using Qiime: you need the sequence data, obviously. You should have quality data for your sequences. You often need to set parameters in a parameters file that we'll be using more later on. And then, of course, you have to have your metadata. So that's all the other data about your samples, what environment they came from, what treatment group, and so on. We usually call that the mapping file. So the sequences file has to be in a certain format. So does the mapping file, and I'll talk more about the sequencing format in a minute. In the mapping file, it's a very prescribed format, so it has to start with this pound or hash sample ID, and then it needs to be tab delimited. So you could make this using an Excel spreadsheet. So the first column is sample IDs. The next column is the barcode sequence for each sample. The next column is the linker primer sequence that was used. Then you can have lots of other metadata like treatment groups, date of collection, and things like that. And then the last column is a description column. And Qiime really likes you to have the mapping file in this format. So you if you want to work with Qiime, you kind of need to get it into this format. If you don't have barcodes in your data, or you don't have linker primer sequence, you can paste something random in there. But you're going to have approximately that format. So those are the various requirements. Then the Qiime also runs off of parameter files. So there are what we call workflow scripts that let you run a whole bunch of analysis steps with a single command. And then you can make a parameter file that will help you tweak the various choices that are made at each of those analysis steps. The key result that comes out of Qiime is the OTU table. And here in the rows are the different OTUs. As a machine learning person, I prefer to have my samples in the rows and my features in the columns, but this is often how it's done in biology. So the rows are the features, and then the columns are the sample identifiers. What it contains is just the counts of each, how many times each OTU was observed in each sample. One point I want to make is that the OTU tables in Qiime are now in a new format called Biome. Biome is not really human readable, but it encapsulates both the count data and also any of the metadata, taxonomy, and so on. And it standardizes the format so that it can make it more easy for one program's output to be used in another program. And if you have a biome file, there are scripts that will easily convert it back to a tab delimited file. So typical Qiime commands for pre-processing the data. I like to run quality scores plot. This will just let you see what the quality of your sequences is as you move along from the beginning of the read toward the end. You can use that to truncate the sequences manually using truncate FASTA qual files. And then if your data are still multiplexed, you can use this split libraries command. That's where it uses the barcodes to separate the sequences. So that's kind of what I think of as pre-processing. Then now you've got sequences all cleaned up and separated and labeled, and you want to pick OTUs and then do all of your downstream analyses. And the Qiime is set up, as I said, to use these simple workflow scripts. So if you want to pick, say, closed reference OTUs, it's one command, and you just give it the sequence files and a couple other things, and then it'll run all of the steps that are necessary for that. Then you can go straight to the last command, which is core diversity analyses, and this will run all of the standard Qiime analyses, produce the nice 3D plots that you can rotate and things like that. Then, so I'll go into just a little bit more detail about the pre-processing step. When you want to do the demultiplexing, you need to pass in the sequence file in FASTA format, a quality file, or you can use a FASTQ file. And you also need to pass in the mapping file, and that's because it's going to use the sample IDs and the barcodes to do the demultiplexing. The goal of the demultiplexing here is to produce a single sequence file with this exact format. The sequence headers have to start with the sample ID and then an underscore and then a different integer for each sequence in that sample. So, it's very important to get your data into this format. And people often get stuck at that point, just getting it to the point where they can feed it into the OTU picking and so on. Once you've got it in this Qiime-ready format, it's very straightforward to just plug it in and get all sorts of great analyses for free. So those are the sequence identifiers there that came from the mapping file. And then the way you get this is the split libraries command, passing in the mapping file and the FASTQ file or the FASTA and qual files. So that was the pre-processing. Then once you've done that, you get to this step of the kind of mid-level processing where you're picking OTUs, generating the features, building a multiple sequence alignment, building phylogeny, making OTU tables, and assigning taxonomy. And this is really the heart of where all of the algorithms that help define the features is. So we're going to be spending a lot of time discussing these different steps, all of the ways that you can do them, why some ways are better than other in certain circumstances. Then once you've got the OTU table and a phylogenetic tree, we can do some diversity analysis. There are two types of diversity analysis that Qiime does. They're usually referred to as alpha and beta diversity. We'll be talking about those in another video. Alpha diversity basically measures the diversity within a sample or within an environment. And then beta diversity measures the diversity between environments, so how different are two different environments. Here's an example of alpha diversity where you would use this script alpha rarefaction, or this could be part of the core diversity analysis. You would pass in an OTU table. You might pass in a phylogeny, because some alpha diversity measures use phylogenies. And then you can get out these nice plots that show you if the diversity is different between, say, different treatment groups. And so we'll be going into all the details of how that works later on. The rarefaction portion is a very important step and has a little bit of controversy, but basically the idea is to find out if you have sampled all of the diversity in your sample, and produces these nice plots where you can see if the diversity that you're acquiring, if you continue to sequence that sample, is leveling off. Beta diversity is another major output from Qiime. So here again, you'd use the OTU table. You would usually provide a phylogeny, because one of the most useful beta diversity or distance metrics uses phylogenetic information. And then you might also have a parameters file. There are several different commands for this. The most common way you would use this is through the core diversity script. And then this produces these nice plots. It actually gives you a web page where you can rotate them around. Here's a list of all of the various diversity metrics that are in Qiime. When you're running Qiime, if you want to get help with any command, you just type the name of the command and then -H. A little hint for non-programmers: if you want to find out how long something takes to run, you can just put time before it and it'll tell you how long it was. I like to do that. So we're not gonna go into a demo of Qiime right now, but I want to give you some references for getting Qiime working. If you have a Windows machine, the easiest way is probably by using a Qiime VirtualBox. So you download this thing called a VirtualBox. It's basically simulating another computer running inside your computer, and it has Qiime installed, everything, all the dependencies and everything. It's just ready to go. If you're using a Mac, I highly recommend Mac Qiime. This is really straightforward. You just download and click install, and then you've got Qiime running on your machine with all of its dependencies. That was developed by Jeff Werner. And then finally, if you're, for people at the University of Minnesota, if you're using MSI, the Minnesota Supercomputing Institute, if you don't have a login, I encourage you to get one. And I have a working installation of a recent version of Qiime, Qiime 1.9.1, and I can help you get that set up. There are several great tutorials for using Qiime online. You can visit qiime.org/tutorials. The first 3 tutorials are especially useful. They walk you through the core diversity analyses, and they also discuss ways of dealing with Illumina data and ways of doing OTU picking.